Now, most of you have probably heard that human and chimp DNA are 98 to 99% the same. Okay, that is the canard I hear all the time in the media, and the question is that really true? And for many years now, evolutionists actually have been saying no. And so in 2000, just to give you one example, this is a 2012 paper by evolutionists that said it is now clear that the genetic differences between humans and chimps are far more extensive than previously thought. Their genomes are not 98 or 99% identical. Now, I'm guessing you haven't heard anything probably about this in the media, because I haven't, right? And you probably won't, because I hate to break this to you, but the media, for the most part, is extremely liberal. Probably all of you already are aware of that. And so they have a certain agenda, most of them, again, not all of them, but they have an agenda, and they want you to believe that you are your own God, right? That you are evolved from nape. And so they're going to continue to espouse those views, even though they're not really scientific, and even the evolutionists wouldn't say that.
What's the comparison of the human number 1 chromosome with the chimpanzee number 1 chromosome? But that's not how they do it. They take the human number 1 chromosome and they search all of the chimpanzee chromosomes to see if they can find little bitty sequences that line up. Doesn't have to have anything to do with it being gene structures or regulatory structures, just little bitty sequences, and see if they line up. That's how they come about with their comparisons.
But you were also saying that there's quite a bit of difference between just the percentage of genome for the chimpanzee and the human.
Correct. There's almost 12% more DNA in the chimpanzee chromosomes than there is in the human.
And so how can you only get a 1% difference when you have 12%?
You ignore the 12%. That's the way you do it. Now, I don't know if the chimpanzee appreciates you ignoring the 12%, because it probably is a little important to the chimpanzee. So that's why in 2010, there actually was a paper that finally did a comparison of all the techniques used to compare chimpanzee chromosome with human chromosome, with fish chromosome, with cow chromosome. And this paper concluded that they're so flawed in all the ways they do this that you get totally different results depending on what method you use and the assumptions of the method being used. And so the paper really suggested that we can't make a claim of what the similarity is. Now, I promise you, they're still going to be claimed 1%, maybe 2%. But we know that you can't make really any claim at all. There's just no way at the moment to adequately compare.
But bear in mind also, it's not like we're lining up gene to gene or functional group to functional group. Here's maybe an analogy that'll help explain it to you even better. Okay, let's take the sentence, "Methinks it is a weasel." Okay, the sentence that Dawkins used so profoundly, tried to use so profoundly in his book, about the blind watchmaker, where he had that pre-programmed random generator that wasn't random and there was no way he could fail. But anyway, so I'm going to use this also, all right? Now let's take the other sentence, "We have the inks to sell." Now, how similar are those sentences? How similar is "we have the inks to sell" to "methinks it is a weasel"? All right, well, if we take the approach that is used to compare the similarity of the human chromosomes with chimpanzee chromosomes, here's how we do it. Here is INKS here. Here is INKS here. Okay? Now, it doesn't make any difference that they functionally don't have the same content in the two sentences. That's irrelevant. We have the I, we have the N, we have the K, we have the S. And that's all we need. All right? Here is W here. In fact, I should have increased it. We have WE here, and here is WE here. Okay? It doesn't make any difference that one is spelling the word we, the other is spelling the word weasel. That's not important. The idea is we have WE and WE. So if we continue to do this, what we're going to find is that, lo and behold, 18 of the 19 letters match. Now, you say, but wait a minute, we have the inks to sell, we have a V and an O that aren't in the "methinks it is the weasel." Exactly, we got to ignore those. Because remember, the chimpanzee has more chromosomes than we have, so you just ignore it, because that's the way the process works. So we're going to just ignore V and O because we don't need them to match up the "methinks it is a weasel." So we already have a 95% match, but we're not done yet. Because see, we can continue, we can take the I, we can bend it, we can make part of the we can use that to make part of the leg of the M. We can take the V out of the W and make part of the M also. So before we're done, see, we can make 3/4 of the M. So what we have in the end is we actually can have 18 3/4 of the 19 letters, and we get a 98.5% total match. That's how it's done. Okay? They're not looking for functional group to functional group. They're not looking for activity to activity. They're just taking, give me a segment, see where it matches up. So when we come back to the picture then with all the different colors, see, here's what you have. You have line up these small segments. But it's not even that tight, because in this small segment where they've lined up, you'll notice it's not a total alignment. There are 2 bases that are wrong, but that's okay, because it's close enough. So that would be considered a total alignment. Okay? We can also have this, but that's okay, because we've aligned all the rest of them. So if we got extra, we just loop that out. See, so depending on the program they use, they can make all these kind of fudge factors, and it's not surprising they can get very close. See, I showed you how to take 2 sentences, nothing to do with each other, and make them 98.5% similar, if I use the right paradigm, if I use the right approach. Okay? So we come back to this, if we take just a very small 1% of the human genome, and this is what it looks like as far as, notice all the different colors. Those colors are telling you that those are the different chromosomes that the program had to go to, had to search through of the different chimpanzee chromosomes. So what we see is in the alignment of chimpanzee to chromosome number 1 of human, we have some from chimpanzee chromosome 1, some from chimpanzee chromosome 2, some from chimpanzee chromosome 6 and 7, etc., because they're taking bits and pieces of it. And notice also we have areas where there's no alignment at all. Even with doing this mixing and matching and this fudging, they still have areas where they can't align them. There's nothing that aligns. No matter how much they force it in, they can't make it align. Right? Also notice, and again, it may be a little hard to see on this, okay? It says chimp chain versus chimp net. What those are is 2 different methods of aligning the chimp chromosomes to human chromosome number 1. If you notice, they don't give you the same color patterns. Chimp chain may go to chromosome 3 and say this will fit on chromosome 1, but the chimp net program says no, I'm going to go to chromosome 6 and align that. See, so it depends on the program you're using. So it really just becomes a matter of, if I can write the program correctly, I can make it 100% match. And in fact, see, that was then brought out in a 2007 paper where they said, you know, we are having to assume evolutionary-based phylogeny. We are having to have problems with, and therefore we have to ignore what are called repeated sequences. We also are taking into account, as in, if we assume an evolutionary history that influences how we align them. We assume human and chimp should be closer, so that's going to be built into the program. We assume human and cow should be further away, that's built into the program. So we're assuming evolution as part of our program. And also, the effects of gaps and missing data are minimized. Okay, there's a gap here, but we're just kind of minimize what that means. It's not that significant. And then, as I said, in even this paper, they acknowledge that at most there's only really about an 87.5% alignment. You don't hear that. Where'd that come from? But here it is in Genomic Research, 2007. And it looks like when all's said and done, what we're really going to get is an 87.5% alignment. And even I would say even that might be being generous.
Now, a creation geneticist, okay, someone who does believe in the biblical account of origins, by the name of Dr. Jeffrey Tompkins at the Institute for Creation Research, has been doing extensive analysis of human and chimp DNA. He's got some major supercomputers there. He's been plugging this information in. And I want to share with you some of his original research. But before I do that, I want to share with you what he found when he evaluated the human-chimp DNA comparisons that have already been done by the evolutionists. He said what he noticed was a lot of preferential and selective treatment of the data, okay? And so, they're not really giving you the whole story, so to speak. So, for example, he found that in some of the papers where they did comparison, they just use sequences that they expect to be similar, like the genes. So here's the problem with that. I would expect our genes to be similar, okay? Genes are what produce proteins, and proteins are what make up our bodies and do all the work in our bodies for the most part. So because chimps are mammals, and so are we, from a biological perspective, of course our genes are going to be similar. Our bodies have to do similar things. If you compare our genes to a dog, a cat, or a mouse, they would also be very similar. But that's not really giving us the big picture, right, of how much similarity there is. Humans, only 2% of our DNA codes for protein. So that's just looking at a very, very small portion of the DNA. It doesn't really give us the big picture. So now what's interesting is they found that when they do compare all of the DNA, that it turns out some of the differences they just don't count. So for example, they're not counting non-aligned DNA, they're not counting gaps, they're not counting copy number variations, and they're not counting size differences. Now, these are legitimate differences between the human and chimp DNA. So why don't they count them? Because they have no idea what to do with these numbers. Because if all of these differences are true, right, and true differences between humans and chimps, there's not enough evolutionary time for that to have happened. See, they proposed that we had an evolutionary ancestor so many millions of years ago that we shared with the apes. All right, so if all of these differences are true, that means they're going to have to give even more time, more millions of years, and then the data is not going to match up with fossil data or radiometric dating data. That's going to be a problem, and all those things have to mesh and come together to tell the same story. So it really messes up their evolutionary timeline. While millions of years might seem like a lot of time to you, in evolutionary terms, that's a blip, okay? It's very, very small. And so to get that many differences, you're going to have more time, and they just simply don't have it. No evolutionary geneticist in their right mind would think that this many differences, so to speak, occurred in that amount of time. And so they know it's impossible. So they kind of ignore it. They don't count it. Now, is that a legitimate thing to do? Well, absolutely not. You can't say, well, those differences aren't important, because you don't know that from an experimental basis. Some of them might say, well, it's just in the junk DNA, okay? It's in the DNA that isn't important. That's the DNA in between the genes. That's 98% of our DNA. It's termed junk. Now, that's a whole other talk I have, by the way. But as it turns out, junk DNA isn't junk, okay? It has a function. It's doing something. So to say, well, those differences are in DNA that isn't important, there's no such thing, okay? All of the DNA is important. All of it's doing something. Almost all of it's functional. So it's going to become an increasing problem for their argument. Now, as it turns out, what they really do count to get that 98 to 99% similarity is they're really only counting one type of difference, all right? So let me show you what they're counting. So when we write DNA, when we write it out, we use 4 letters, A, C, T, and G, that are the 4 bases of DNA. And what we find happening is that in regions that are very, very similar, which are called the alignment regions, there are places where there is human DNA, which we'll say is the top line here, and chimp DNA, which is the bottom line here, but they're not the same. Okay, so those are the only differences they count. Now you can see there's a difference here, right? There's a gap. There's another gap here. This is what they would call non-aligned DNA that doesn't match up. Okay, so why aren't you counting those differences, right? But so that's clearly a problem. And again, it doesn't fit their evolutionary story, so they don't count it. In my opinion, this is very bad science, and it's very deceptive science, because it's not really giving you the true story of what's going on. So what's the reality? Okay, how many differences are there really between human and chimp genome? And Dr. Tompkins recently published a paper on this, and he found that there's probably only about 88% similarity overall. Now, that might sound like, well, that's still a lot of similarity, but you got to consider that's a 12% difference, which 12% of 3 billion, you're talking 360 million differences. That's a lot. Okay, a lot of differences. And no geneticist, again, in their right mind is going to believe that that many differences occurred in that many millions of years. I'm also giving them, for the sake of argument, that mutations do what they need them to do, okay, so that the changes in DNA can lead from one kind of organism to a completely different kind of organism. As it turns out, mutations don't do that, okay? So even if they could have all these differences, it wouldn't matter because mutations just don't do that, okay? We can observe that and we can see that that doesn't happen. So their argument fails on many, many levels. Now this does not count, this 88% figure does not count the fact that chimps have an 8% larger genome than human beings. So if you count that in, then there's only about an 80% similarity. And this doesn't count the fact that you're just talking about the linear sequence of DNA, the string of bases, so to speak. DNA is a three-dimensional structure, and we're still figuring out all the layers of information that are there. For example, epigenetics, that's an area of genetics where they look at the chemical markers on DNA that affect what we call the expression of DNA. It's very different between humans and chimps. So this is a very conservative number. The reality is it's probably much lower than this as we continue to figure out how the information is encoded in the DNA. So there's no way to account for this many differences in the evolutionary time frame, leading Dr. Tompkins to conclude that this defies standard evolutionary timescales and dogmatic presuppositions about a common ancestor. There's just no way. There's too many differences.
In 2005, which is, as far as I know, the most recent chimpanzee alignment, when they sequenced the chimpanzee genome, it wasn't a matter of just sticking it into a sequencer and just churning out all these millions and millions and millions of bases. You have to piece it together. What they did was they pieced it together using the human genome as the scaffold. Because the assumption was, well, human and chimpanzee are so genetically similar, it ought to very easily line up. Any competent geneticist now knows that 2005 chimpanzee alignment was a disaster. Well, if evolutionary predictions were so brilliant, it shouldn't have been such a disaster. But that's exactly what creationists would have predicted, is that's going to be a disaster. Who was right? We were right. Then you got to figure out, okay, this segment fits here. So this segment starts at, nucleotide 1752 and goes to nucleotide 2717. And so you got to figure out how to set the segments together. Well, when you're working in the dark, you don't know where to piece these together. So that's why they use the human as a scaffold to build it on. When they realize that doesn't work, okay, back up, put the chimpanzee together on its own. That's what they're trying to do, and it keeps falling apart. So they don't, that's one of the reasons, if you notice in the video, when it was saying, when I made the comment of the 12% larger chimpanzee genome, that was based on one paper. See, it varies. What's the actual size of chimpanzee genome? You'll never get a consistent view. Why? Because they don't yet have it lined up. In fact, you don't even get a consistent view of what the size of the human genome is because it keeps changing some too. So how do you know it's only 1% difference when the size of the genomes keep changing?
If you took the human genome, and oftentimes the comparison is done to how often would you expect to find this in a random sequence. If you took the human genome, chromosome number 1, and compared it to the same number of random nucleotides that you would find in the chimp, can you find, and especially if you're looking for just little pieces, 15 base pairs, you're going to pretty often find that in a random.
You will. That's a very good point. Yeah. If you just randomly generate a sequence, and in fact, I almost think that somebody might have done that. You might be able to point me to the paper, but I'm almost thinking, a recess in my mind, somebody actually did that and said if you randomly generate a sequence and then use one of these programs, lo and behold, you still get some matching with the human genome to this randomly generated sequence.